All right, so in this video we want to look at how to quantify the number of lipid droplets per cell. Okay, so we're going to work with this particular image of lipid droplets. And the first thing you want to do is just like with the area, if you're doing area 2, is you want to identify which cells you want to use. And sometimes finding the cells are really hard. So how are you going to find cells? Sometimes the best way to do that is to go to your image of all the cells. And so this is the merged image. And sometimes the merged image, you can see the nuclei, you can tell better. So to identify the cells, what we want to do is we're going to go through this multi-point selection tool here. Okay. So we're going to click on that, and then we can click on and identify. So let's do this cell, we'll do this cell, 2, 3, 4, 5, 6. I don't know, maybe we do 7 cells here. And that might be enough, for quantization purposes. So maybe let's do 7 cells. And so what you'll do is then you can save this particular image, right? So come here, File, and then Save As, and then save this image and make sure that these particular selections are saved so you know exactly which cells you're going with. When you're selecting cells, do not select cells on the edge or the periphery of this panel. And you want to select cells that have distinct puncti in them, distinct lipid droplets. If there's a big blur of them, that's going to be really hard, especially in this particular example of phenotype where you're counting the number of the spots per cell. So there is going to be a little bit of a bias there, but that's just because of the limitations of what we're trying to do. So once again, maybe pick 5 or 6, 7, something like that number of cells, depending on what you're trying to get at in terms of quantization. You may have to go back and count more. I don't know. So then you've selected those, you save this image. If it doesn't save for whatever reason, you can always just do a screenshot of this so that as long as you recall which ones are which. And this is great information to have. You want to save this in your records in case you have to go back and recount for some reason, which has happened in the past. So something to think about. So then you would go into, let's see, we're going to the stearic acid. So this is the lipid droplet only image. This is where we want to be. And what we're going to do is we want to zoom in on the individual cells. Now, once again, for the purpose of this class, I counted 1, 2, 3, 4. So you would just count. You start with the first one, and you're going to do one cell at a time, because ultimately the number you want to get, the phenotype you're trying to measure, is number of lipid droplets per cell. Okay, and so we want to do multiple cells in order to get that. So let's just say that this was 1 down here, even though I know it's not. We want to zoom in on that one cell, and so you can use this magnifying glass here, and you can zoom in on this one cell. And let's just say this is our first cell, right? And so if you don't remember where one cell begins and the next cell ends and so forth, go back to the original merged image, right? And go from there. Now, for this particular purpose, there's a couple of different ways one can do this. I'm going to show you the simple way. If you want to go into more detail, I would suggest going on Google and typing in a variety of ways to do this as well. Google image search with ImageJ is just really intuitive. It works really well. But for our purposes, the simplest way to do this is to use this multi-point selection tool and make sure that it has numbers on it. And you may have to, every time you do a new cell, you may want to adjust the values, etc. like that. But this is pretty straightforward. So we're just going to select a point, and that's 1, 2, 3, and it keeps track of it. So if you get tired and you need a break, right, you're just going to go through. And you can see how I'm giving all these singles where I can see distinct lipid droplet differences. And so far this has been pretty easy, right? Pretty easy. All right, and then these, and you might have to zoom in deeper, right? And that's okay, right? So if we want to zoom in deeper, and we're like, I really don't know if that's a spot, if that's a location or not, so we can zoom in even deeper, right? And you can start distinguishing what's a lipid droplet and what's not. And sometimes it's a little easier than others, but I would classify that as 2 there. And then, at this level, we can start distinguishing, what's what here. Now, what to do about the blobs? All right, this is where it gets a little bit interesting. And so what I would do is I would purposely try to, equally in every sample. So if you have 2 different or 3 different conditions that you're comparing, I would try to be equally biased in whatever you do. So if you're selecting cells and you're doing this measurement and you want to select number, make sure that on each one that you're equally biased. So let's say, maybe I wouldn't even pick this one if I saw this, right? Maybe I just picked one that had distinct punctae all the way through, and I would do that for all of them on this screen, and I would do that for all of them on the other conditions as well. But if you have to do something like this, here's what you have to do. I always go with the bright spots as my lipid droplets, and then it just gets a little bit absurd, right? And there's just no good way around it. So you would have to do something like that. That is not ideal, obviously. And so my ideal is to preferentially bias a little bit what you're doing there for those lipid droplets to have more like this. So if we're going over here, maybe we can zoom in over here just so I can show you what I mean. So, this one over here, the lipid droplets are a little bit more manageable. This big blob here in the center is actually a different level of a cell. So it's not actually even the same cell, so it's not even distinguishable. So, like some cells, like down here at the bottom near the scale bar, are going to be easier than the ones on the outside. So you have some situations like up here, there's definitely some that are more distinct that I would use compared to this one over here, and definitely the one that we picked. But that's how you do it. And then you just get the total number there per cell, and then you add that value to your Excel sheet, and you're going to work that up in cells. So then you move on to the next cell. And so how do you refresh all this? Quite honestly, the easiest way is just to, exit out of this, don't save, and then reopen it again. And then by it sounds a little silly, going through all this, but by reopening it again, then, you can start fresh. You don't have to worry about weird things. But then you know exactly which cell you're going to next because you have that on this merged image. You're going to know exactly where you're going to next, okay? So that would be how you would quantify the number. And like I said, how many cells you choose is kind of up to your group. There's no such thing as too many. It's just amount of your time and how valuable it is to you. But I would say definitely 5 to 10 cells. Usually 10 cells is normally pretty good on this particular example.